Okay, so here we have our second in the lecture series on cell transport. This is on osmosis, and this is a concept you should be familiar with from middle school. We're going to be talking about the movement of water. Here's your list of objectives. Read over them, make sure you understand them by the end of the lecture, and if you have any questions, you can ask your teacher or you can email them. So osmosis is literally the diffusion of water. It is its own separate category because of how important water movement is to a cell. The water is going to be a polar molecule, and we know that from looking at the properties of water. However, in this case, it is completely permeable, meaning able to pass through, because the cell membrane allows it to fit because it is small enough. If you think about the size of these phosphate heads here, this phosphate head has at least one phosphorus with 4 different oxygen molecules attached to it, and a couple other side groups as well. A little hydrogen, 2 hydrogen molecules plus the oxygen. That little water molecules can be very small. It will easily be able to slip through and fit through without having any problem. So diffusion of water is what osmosis is. Water is very important in helping us to maintain a balance in the solute concentration inside and outside of the cell, because the cell is always trying to reach equilibrium with its environment. But sometimes solutes can't move. And what happens then? Well, here we have a picture. Let's look at this. We have these large blue solute molecules, and they're not able to cross the membrane. We have a higher concentration of solute on this side, and then we have our membrane here, and a lower concentration of solute on this side. Under normal consideration for diffusion, what would happen is our solute molecules would move from high to low. But look at how big they are. They're not going to be able to move. So now we have to look at the ratio of how much solute to water there is. So here we have lots of solute and very little water. Okay, let's count them up. We got 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so on this side I have 9 solute particles, and on this side I have 3 solute particles. All right, but then as far as my water molecules go, which represent the red, I have 1, 2, 3, 4, 5, 6, 7, 8. Okay, there are 8 water molecules, whereas over here, I have 18. Okay, so if we look at that concentration, there is much more solute concentration here than here. So if the water is able to move, could we balance out its concentration? And the answer is yes. The water molecules on this side are going to diffuse across until we have an equal concentration. It's sort of like saying, let's change these numbers. Let's say we had 90 particles of solute in 100 molecules of water. That would be the same thing as 9 particles of solute in 10 molecules of water. You see how they're equal to one another? It's about their concentration, and the concentration is the amount of solute per solvent. So that's really important to understand. It's not about the number of molecules, it is about the concentration. So water is going to continue moving until the concentration reaches equilibrium. So we might have more solute particles here, but we're going to get much more water flowing to this side to balance out our concentration. I know it's an abstract concept, and we'll work on it some more in class, but I think that this might help explain it the best. So keep this in mind. Okay? So solute is unable to move, but water can move to help us create an even balance or equilibrium with our environment. So let's take a look at different possible solute concentrations inside and outside of the cell. Here we have 3 diagrams, and we're going to look at them each in a different slide. There are 3 different tonicities. Let me get that word up there for you. Okay, tonicity, that is referring to the amount of solute concentration. So sometimes the solute concentration is lower outside of the cell compared to inside. So hypotonic, hypo, and low, that's what I think of. Hypotonic means that there's less solute outside of the cell. Here we have very low solute concentration, and inside of our cell is a very high solute concentration. So what's going to happen to the water that's able to diffuse? Water is going to move into the cell. Okay? Just remember, this is a good tip here. Water moves to wherever the solute concentration is highest. Okay? It always wants to balance things out, so when it sees a high solute concentration, it's going to naturally diffuse that way. Because if there's a high solute concentration, what does that mean about the amount of water in that area? It's at a low concentration. So water is going to move from its high to low concentration. Okay, right? See much more water here, high concentration, and it's going to move to its low concentration. That's going to be the opposite for the solute particles. Okay, they're not going to be moving at all in any way. So let's take a look at each of these. We'll go through hypotonic, isotonic, and hypertonic. They each get their own slide. Okay. Remember, cells are trying to maintain internal conditions. So isotonic, iso means same. That means the concentrations are already balanced. So the amount of water that is entering the cell is equal to the amount of water that is leaving the cell. So we consider this at equilibrium. Does water stop moving? No. Okay, that's an important thing to understand. Water molecules actually never stop moving. Even once they have achieved this balance, there's still going to be small movements. And so what's going to happen is the cell is going to react very quickly to keep that homeostasis going on. An example of homeostasis is in your blood. Okay, your blood has a 0.9% solute concentration. So it's very, very low. It is made of mostly water, right? And that's a good thing because it's a fluid, it has to flow. So your blood is always that 0.9% concentration. Whenever you go into the hospital and they hook you up to an IV, they can't just give you straight water. That would be terrible. Like let's say you got dehydrated and you passed out and they needed to give you some fluids. If they gave you straight water, that would be 0% solute. And even though it seems like it's not much of a change from 0.9%, it is a serious change. So whenever they give fluids to a person intravenously, that bag of IV solution contains a 0.9% solute concentration so that the concentration is going to be the same and it's not going to cause any significant change in the movement of water. So the molecules will always continue moving. So even here, when we have received balance, right, we have an equal concentration of solute and water here, equal concentration of solute and water here. What's going to happen? Let's say this water molecule gets enough energy and goes shoom and comes across this way. Well, this one in immediate response is going to move into the cell, right? And that brings it right back to equilibrium. So we're at equilibrium. This is an isotonic condition. And an example of that is in your bloodstream when you're receiving fluids. Okay. Sometimes the concentration outside of the cell is lower. This is that hypotonic that I started talking about. Because of that, we're going to see water moving into the cell in order to achieve balance with the outside environment. So what's going to be the response of that? That's going to cause cells to swell. They're going to fill with water more than what they normally have. Okay, their usual concentration of solute is going to change because it's trying to match its outside environment. So water is going to diffuse into the cell where there's a higher concentration of solute, and that's going to cause it to swell. And this can be a really bad thing. Remember what I was saying about the IV transfusion? If you are receiving straight water, 0% solute, that's going to be a lower concentration compared to what's in your cells, or I'm sorry, what's in your bloodstream. And what's going to happen is it's going to cause water to move into your bloodstream to balance out that difference, uh, and it can cause cells to swell to the point that they will burst. And those are your red blood cells that are in the bloodstream. So they're going to take on this extra water, and all we have is a thin, flexible cell membrane wrapped around our cell contents. That's very, stretchy to a point, but if the cell fills up with too much water, it's going to pop. Okay, and we have seen some terrible conditions of this. I don't know if you've heard in the news within the last few years when the Nintendo Wii came out, there was a radio competition that was called Don't Wii for a Wii. And basically what they did is they asked their participants to drink gallons of water and not relieve themselves by going to the bathroom. And so what ended up happening is that their cells became in a hypotonic state, and they were taking on too much of the water that was being put into their bodies, and they weren't able to get rid of it by excreting it in their urine. And the result is that they died. Okay, so maintaining a stable internal environment is very important. Hypotonic conditions for animal cells are very dangerous. Okay. Let's look at the opposite effect. What if it's hypertonic? Okay, hypertonic means that our solute concentration outside of the cell is greater than inside of the cell. So look at all these particles here, all these solutes. Again, they're not able to cross the membrane. So we rely on water to help us achieve that balance of concentration. Water moves wherever the solute concentration is highest, so it's going to move out of the cell. Well, what effect is that going to have? That's going to cause the cell to shrink or to shrivel up, and we know water is essential for a lot of cell functions. If most of the water is naturally moving out of the cell through its diffusion property, through osmosis really, then that's going to impair the cell's function. And so we can see this in a little, toddler, young child experiment. You have a slug, and that slug has a very high concentration of water compared to the air, which has a very, very low concentration of water. In fact, there might be some water vapor there, but there's nothing liquid. Okay? You make it even worse for that slug when you devilishly pour salt on top of the slug. Okay? Anyone who has done that will know that as you pour the salt on top of the slug, what has happened? You have created lots of solute molecules on the outside of that slug's cell membrane, and so it's going to draw water out of the cell, and it's going to literally cause that slug to shrivel up, and that's going to cause the slug to die. Sorry, there's a lot of death in this lecture, but I hope you understand that maintaining equilibrium is very, very important to our cells. So as I said before, in non-isotonic conditions, animal cells are going to be threatened in both ways. So here we have a picture of red blood cells, and actually if you go on the website under the A4 unit links, you're going to see that there's a link that shows you what happens in real life to red blood cells that are in a hypotonic condition and a hypertonic condition. So here we have normal, regular red blood cells, and they are very important to our bodies because they help us carry oxygen. And that is what circulates the oxygen from our lungs to all of the cells where we need them in order to use it for cell respiration. If we put those cells into distilled water, okay, this is an example of a hypotonic environment. The concentration is lower outside of the cell. It's going to cause water to rush into the cell, and that's going to cause the cells to swell and most likely burst. This is called cytolysis. On the other side, we have a hypertonic solution. Let's say that we increase the solute concentration outside of the cell. That's going to cause water to move out of the cell, and that's going to cause them to shrink or shrivel, and this is called crenation. Both of these, do you think these cells are able to carry oxygen very well? The answer is no. So the cell loses its function, and in an animal cell, that can be very, very detrimental. Plant cells are a little bit different because plant cells have a cell wall. And the cell wall acts as an extra barrier of protection. So the cell wall plays a role in keeping a plant from bursting. And because of that, that vacuole, we like to fill it up. Well, what's one thing that we fill their vacuole with? Water. Most of it is filled up with water. And I think in an earlier demonstration when we were talking about vacuoles, I explained it like this. Imagine the vacuole is like it's like a water balloon, okay? And you can continue filling water in that water balloon, but eventually it will pop, okay? That's like an animal cell. Instead, take that water balloon and now place it inside of a sturdy cardboard box, okay? And that box doesn't have much flex to it. Now as you start filling up the balloon with water, it's going to stretch and push, and what ends up happening? The walls of that cardboard box start exerting a pressure back onto the vacuole. So essentially what's going to happen is as the vacuole starts pushing out towards the cell wall is going to put pressure back on it. And so what they're going to get is they're going to get this limit. They're going to reach equilibrium when the vacuole is extremely full. So what ends up happening for a plant cell is the cell is going to stiffen, right? It's going to stay very turgid. That's the term for this. It's going to stay very turgid. Oops, that's an arrow. But generally, it's not going to burst. It's going to maintain its shape. The cell wall is going to prevent it from bursting. Whereas if you put plant cells in a highly concentrated salt solution, so this would be an example of a hypertonic solution, it's going to cause something called plasmolysis, and that's going to cause the cell membrane to shrink away from the cell wall. Remember, the cell wall is very sturdy, and that's not going to be moving in any way. Instead, your cell membrane is going to pull away, and you're going to see your plant shrinking. We know how water is very important to plant cells as well, so plasmolysis would be a very detrimental condition for plant cells. So vacuoles, as we said, they store water, and you get this osmotic pressure, which is going to allow for the vacuoles to fill up very full, but yet not burst the cell as it would in an animal cell. And when the vacuoles are very full, they are happy, so hypotonic conditions are good. When they're empty, we can tell that because literally the structure of their cells become wilted, and that's what we see when a plant is starting to experience an isotonic or a hypertonic condition. So the desired concentration for an animal cell is to be isotonic with its environment, right? Here's normal, okay? So for the animal cell, normal is right here, but for the plant cell, normal is going to be right here. So it likes to stay turgid in a hypotonic solution, whereas animals like to stay isotonic at balance. Okay, so why is this important? We experience this on a regular basis, okay? You take a bath and you decide you're going to sit in there and lounge, maybe read a book. The bath water is very hypotonic compared to you. You're about 70% water, which means you're 30% solute concentration in the majority of your cells. The bath water is complete water. That means that it's 0% solute, although I would say it's not pure water because there's, some ions and some other metals. There's fluoride in it as well. But for all intents and purposes, no solute. So what ends up happening? Water is going to start diffusing across your skin cells by osmosis. Okay? Don't fear. It's not like next time you take a bath that you're going to explode because your cells are filling up with water. Our skin has a very protective waxy barrier on the outside that does some waterproofing. But if we sit in that solution for a very long time, what happens to your fingers? They get those wrinkles. And what's happening is the shapes of the cells on the ends of your fingers are changing, and it's causing these wrinkles to form because they are filling up with water, and it's causing them to have a different shape. We also can see this at a grocery store, okay? Vegetables generally are being shipped. They might spend a day or two on a truck before they get to your grocery store. And I know, ew, that's kind of gross to think about, but it's okay. Everyone does it. So when those, let's say beautiful strawberries arrive from California, they might be a little not good looking because in the trip they lost some water to their environment. So what ends up happening is the grocery stores have those little spray systems set up, and like if you've ever been shopping and you notice they'll mist water, every like 2 minutes or so. Why are they doing that? Because the water falls on the surface of your fruits and veggies, and it's going to cause them to plump up. So okay, and why does it do that? There's a hypotonic condition now. There's water in greater concentration on the outside. In other words, there's a low solute concentration on the outside. So those cells are going to take the water into their cells, and that's going to cause their cells to fill up more with water. But that plumps them up, so then they look like they are nice and fresh and juicy, and that's exactly what we want from our vegetables. So those are just some examples. We're going to be doing a really neat lab where we're going to test some different effects of osmosis on a very familiar cell to you, an egg. Okay? See us in class.